Okay, gang, we are here at section 10.3. Here's your video about the Calvin cycle, and I am looking at this diagram from the book, and I don't know, it seems really, really straightforward to me, so I'm not 100% positive where the questions would be on this, although as I look at it, I feel like it's really confusing, so I'm not quite sure what to go over. So I think I'm gonna do two slides here. One little section where I just go over exactly what the diagram says, and just point out the key points, and maybe that's all straightforward to you, and there's no confusion. And then I'm going to do a second one where the one thought that I had was, as I look at the diagram, the pictures aren't very detailed about the molecules themselves, and I know some of you often have questions where you would want to know what some of the differences are in these molecules because they look exactly the same the way they have them depicted here. So I think I'll draw on some of the details, which might answer some of those questions. But anyway, so first of all, the Calvin cycle here, it has three different phases. Up here there's phase one, the carbon fixing or fixation phase. Fixing carbon is just taking carbon out of the environment or out of the air and putting it onto an organic molecule. So that's the big point of phase one, just take carbon out of the air, put it into the organic molecule. Phase two is the reduction phase, so we're going to be adding electrons to something. If it was oxidation, we'd be taking the electrons away, but we're going to add the electrons, so it's reduction phase. And then the last phase is to basically get back to the starting point of the cycle. So if we look at, let's just start from the beginning here. When we look at what we start with something called rubulose bisphosphate, and RUBP, RUBP, and that is our starting material, and we're going to take one of those, and we're going to add carbon to it. Okay, and this is the carbon fixing stage, so we're going to take a carbon dioxide and add that to our RUBP. They highlight here Rubisco, that's the name of the enzyme that does this. I think the only reason they highlight it is, as the book says, it's probably the most common enzyme in the entire world, this Rubisco stuff. But, sorry, I'm going to rearrange here. Anyway, okay, one of the things that would be confusing, I think, about this diagram, they show all the pictures once, but then they have a little number beside them saying it's happening more than once. So we've got the rubulose bisphosphate. We're actually going to take three of those, and we're going to add three carbon dioxides, one carbon dioxide to each of the RUBPs, or the RUBPs. And we get this thing they call a short-lived intermediate, but what we did right then is we did carbon fixation. That's what we did. We fixed the carbon to an organic molecule. So right now we have three molecules. Each of the molecules has 6 carbons on it. Yeah, what we're going to do to those next is we're just going to split those in half. Okay, so the next thing we get is if we split 3 molecules in half, and each of those molecules had 6 carbons, then now we're going to have, instead of 3 molecules, we'll have 6, so we doubled the number of molecules, but we have half the number of carbons in each. So again, 6 of this next thing called 3-phosphoglycerate. Okay, that 3-phosphoglycerate, we have 6 of them again. What we're going to do is add a phosphate to it, to each of those, which will actually make it a little bit chemically unstable. But anyway, so we're going to use 6 ATP. That's going to be adding 1 phosphate to each of the 6 phosphoglycerate molecules, 3-phosphoglycerate, and that's going to make something called 1,3-bisphosphoglycerate. The names are not important here at all, but that you understand that we added a phosphate to each of them. So we have 6 of these molecules, and then we're going to take each of them and we're going to add some high energy electrons to it. And essentially those electrons and the hydrogen that is attached to them, they're going to be attached to the terminal or the end phosphate that is shown here on the left. That's where those electrons are going to be attached, as well as their hydrogen. So it's going to kick out the phosphate that we just added. Actually, we're going to kick it out of there. And that is the reduction step. Okay, so we just reduced it by adding electrons to it, and kicked out a phosphate. So now we have a molecule called glyceraldehyde 3-phosphate, G3P, and this is actually what we want. So we'd have 6 of these molecules, these G3P, glyceraldehyde 3-phosphate. If you guys didn't have to, but if you happened to memorize glycolysis, all of these molecules that they're naming here so far, 3-phosphoglycerate, 1,3-bisphosphoglycerate, and glyceraldehyde 3-phosphate, they're all molecules that were a part of the glycolysis of sugar, the initial breakdown of sugar. Anyway, so we have this glyceraldehyde 3-phosphate, G3P. we're going to take one of those. So there's 6 of them that we made. We're going to take one of those and remove that, and that technically is sugar. Okay, and it can be used. It's a type of sugar. It's not our monosaccharide, but it's a type of sugar, and it can be used to make a whole bunch of other things. Probably the most important that we would think about is glucose. So we can take one of those, and if we did the cycle again, made another one of those, we could put them together and make glucose. Okay, and that's kind of the goal in this whole thing. But so this G3P. So again, we made 6 of them. One of them is being taken to be used to make sugar. The other 5. We need to take those and use them to regenerate our starting material so the cycle can continue. So that's what we're going to do. This regeneration phase is going to take, and if we just do the math on this, we have 5 molecules of 3 carbons. So we have 15 carbons here. We're going to go through a whole bunch of steps. One of those steps is going to take phosphates from ATP and add them to these molecules. But anyway, we're going to take these 15 carbons, rearrange them so that we end up with 3 molecules that each have 5 carbons and a phosphate on each end. We picked up these phosphates from the ATP. Anyway, and that was our starting material. Okay, our RuBP. That's our starting material so that we could do this again, and could do it again and again and again and again and again. We have the cycle going on here. Okay, so I feel like I just said what was on the diagram, like pretty straightforward, but I know sometimes the numbers of carbons can kind of mess people up here. I think that it might, but anyway, the next thing I'm going to do is I'm just going to write a couple things on this picture. I'm going to pause here, write a couple things on the picture, and then I'm going to start back up, and I want to explain specifically here, if we look at the 3-phosphoglycerate and the glyceraldehyde 3-phosphate, they look like the exact same molecule the way they have them depicted here. So I just want to show the difference between the two. Maybe it'll help you understand what's going on with this oxidation or the reduction step and all of that. So, okay, here I pause it. And now I hit record again, and I just drew in some pictures here, or some diagrams. Again, if we look at this glyceraldehyde 3-phosphate and compare it to the 3-phosphoglycerate, their pictures look exactly the same, and I knew that I just thought that might bring up some questions for some of you guys. So if I look at the 3-phosphoglycerate, there's actually a ton of stuff hidden in the name. Organic chemistry, which is what this is, these diagrams, is really, really fascinating, I think, the way that chemists have come up with ways of describing and naming things. But anyway, I'll explain this name actually. The end of this, glycerate, glyc means 3 carbons, so you'll see this recurring thing of glyc in here, means 3 carbons. Glycerate, when it ends in ate, it actually is telling that we've got this double bonded oxygen here to the carbon with another oxygen attached to that same carbon, and that's the 8 part of this glycerate. And the 3, actually, if we number the carbons, which is how they do organic chemistry, they number the carbons, we would say number 1, 2, and 3, the phosphate is attached to the 3rd carbon. So we have the 3rd carbon with the phosphate attached to it on a glycerate molecule. Anyway, that's how we name it. The 1,3-bisphosphoglycerate, we've added a phosphate to the the 1st carbon. So now we have a 1,3-bisphosphoglycerate. Okay, we still have the glycerate, we just added a phosphate. Anyway, the difference between the glyceraldehyde, that it changes the end of the name, glyceraldehyde, and an aldehyde group is minus the oxygen up here. So when we remove the phosphate in our reducing step, we are breaking this bond right here between the carbon and the oxygen, replacing it with some high energy electrons and then bringing the hydrogen in with them. And so the glyceraldehyde 3-phosphate, here is where the energy is stored, and these high energy electrons that would be associated with this hydrogen is where they're bonded. So that is that reduction step there, and that's the difference between those two molecules. And again, this glyceraldehyde 3-phosphate, that's one of the things we want. That's the big thing we've made. We've just made sugar, hooray! With our ATP and our NADPH from the light reactions. Is how we made this. So I'm going to end it there. I don't know if I've done the Calvin cycle justice or not, but hopefully this maybe eliminated a question or two. And we'll see you in class.